Hey guys, Mr. Hyatt here. This is the Chapter 13, first part of Chapter 13 lecture over meiosis and sexual life cycles. So, of course, we're going to start with several vocab terms. Hopefully these are reviews from past courses that you've had. But we need to have a foundation of, genetic vocab built up. That way we can apply some of this stuff, certainly in second semester when we come back and we start learning about Mendelian genetics and non-Mendelian inheritance and things along those lines. But start off with our definition of heredity. That's going to be anything that can be passed from one generation to another, from parents to offspring. Notice we've been aware of heredity for thousands of years, much longer than we've been aware of genetics and how that mechanism works. Genetics is a scientific study of heredity, so how traits are passed from one generation to the next. It's only about 150 years old. Really, Gregor Mendel launches us in the world of genetics in the 1860s. So another foundational term here, variation, is going to be differences. Essentially, any difference between two organisms are called variations in terms of genetics. We're looking at phenotypes typically, but eventually we'll get to where we'll look at genotypic variation. Offspring only resemble their parents and siblings. They're not identical matches to their siblings. That's what that point is supposed to make. Genes, remember our definition, Mr. Hyatt's working definition of a gene is something that does something. That's a stretch of DNA that codes for some sort of protein, codes for some sort of a trait, something along those lines. Locus is called, think location when you see that. That's the address of the gene, where it can be found, which chromosome number, which arm, which length, all that kind of stuff. Okay, when we define reproduction, we're talking about how we copy genes and pass them on to the next offspring. So when Mr. Hyatt says reproduction, this is what he's talking about. Nothing else, just how we copy genes. We organisms copy genes and pass them on to the next offspring. We've learned a ton about asexual reproduction already. That's basically mitosis, also known as cloning. There are quite a few organisms that reproduce asexually. One parent passes all of its genes on to its offspring. So a couple of examples here. Redwoods reproduce asexually. Hydra reproduce asexually. You see we've got a bud coming off of the parent. It's essentially like the hydra grows an arm. Then there's a pinching that happens here. This bud pinches off, floats away, roots nearby, and becomes a new hydra. So again, all of the genes go from one organism to the other. Here we've got SpongeBob, and I know you can't hear the audio, but SpongeBob reproduces via budding. SpongeBob's a sponge. He's saying, can you reproduce through budding? Can you? Can you? Can you? So, I don't know if you know this or not, but they actually staff marine biologists to try to make SpongeBob as sciencey as possible. That's kind of obviously a silly example of asexual reproduction. Advantages to asexual reproduction: you only need one parent. I'm awesome. My kid gets to have all of my awesomeness. So all those great genetic traits are conserved and reproduced. I don't have to worry about some of my awesomeness being recessive to my wife's unawesomeness or something along those lines. I think you get the idea. Disadvantages: you don't get new DNA combinations, so there are no new traits for evolution to work on. And so if I'm really well fit to my environment and I asexually reproduce, my offspring is going to be really well fit to that same environment. But if the environment then changes, my offspring is in trouble. They're not going to be able to adapt because there are no new traits to work on. So sexual reproduction, that's what we're more familiar with. That's where two parents are going to contribute DNA to one offspring. Most organisms definitely reproduce this way. We don't know for sure in some fungi and things along those lines. Most organisms certainly reproduce sexually. But like I said, there are several examples of some that do not. So in terms of advantages of sexual reproduction, the offspring is going to have a unique combination of DNA, which may be an improvement over both parents, may not be. The new combination of DNA is definitely going to be great for evolution because there's going to be new traits to work on. Of course, there are disadvantages. You need two parents. You can lose, like I said, in the sexual reproduction chunk. I'm awesome. I could lose some of my awesomeness that my kid may not get. The offspring may be less fit than the parents. Of course, it requires specialized structures. The parts have to fit together to trade the DNA to make the baby. And then finding a mate is energy intensive. Sometimes you have to be intrinsically attractive to the opposite gender. You have to have traits that they find desirable, and sometimes you have to perform specialized behaviors, and we'll pick up with this in video 2. Okay, so these two questions really launch our discussion of meiosis. Do parents give their whole DNA copy, so their whole genome, to each offspring? Of course, the answer to that is no. The chromosome number would double each generation if it did. So our chromosome number is essentially constant for a species. So we say humans have 46 numbers. These are diploid numbers of chromosomes. So that's a 2n equals 46. Corn has 20, onions have 16, dogs have 72. If you have more than 72, or if you have 71 or 73, you're not a dog. If you have 44 or 41, you're not a human unless you've got some sort of a disorder that throws off your number of chromosomes. Here we see our sexual life cycle if we only performed mitosis. So let's start down here where humans, there's a human male, a human female, goes through mitosis, makes a sperm cell that has 46 chromosomes because mitosis only makes clones. Those two gametes fuse to form a zygote that has 92, which is now no longer human. So our chromosome number would double every generation, so we must need a method to reduce that number. So we go through this process called meiosis, where we take our 46, cut it down to 23. Females do the same thing, donate to the zygote. We get the correct number of chromosomes in our offspring. I use that term gametes. Meiosis is used to produce these gametes, which are sex cells or spores. The purpose of meiosis is to reduce the chromosome number by half, which in effect prevents the chromosomal doubling each round of reproduction. So a sexual life cycle is going to have alternations of meiosis and fertilization and mitosis to keep chromosome numbers constant. Here's what we're talking about. In a human, we go through meiosis, we create gametes, gametes fertilize, then we do a whole bunch of mitosis, and the next generation continues the sexual life cycle. Goes through meiosis, fertilization, whole bunch of mitosis, then eventually meiosis, and so on and so on. Another vocab term here, ploidy, is going to be the number of chromosomes in a set. So N is haploid. That's our number of chromosomes per species. So in humans, our haploid number is 23. Okay, so diploid is going to be two sets of chromosomes. Di, of course, for two, and ploid from ploidy. So that's our most common number of chromosomes in our body, or our somatic cells. Our diploid number, of course, is 46. Corn's diploid number is 20. Notice that 2n number there. Fruit flies are 8, which is really important when we get to learning about Drosophila and all the genetics that come from that, but we'll talk about that later. So human chromosomes, we know a little bit about karyotypes because we've done a lab on them already. Human somatic cells, so all of our body cells that aren't directly involved in making babies, have 23 pairs of chromosomes, so our diploid number is 46. The two chromosomes in each pair are called homologous chromosomes, or homologs. Chromosomes in a homologous pair are the same and carry genes controlling the same inherited characteristics. They're the same length and carry not exactly the same genes. One came from mom, one came from dad, but they're very similar. Here you see a rough cutout of an old school karyotype. This is an old picture that we used to get. We would wait until the cell was at a certain phase of mitosis, which you should know, when the chromosomes become visible. Then we would basically just freeze frame it and then we'd have to cut them all out and arrange them and put them in order so that they looked all nice and neat like this. We used our length of our chromosomes and our banding patterns to figure out what matched with its pair. Notice this is a human female. We can tell by the two X chromosomes in the bottom right. Again, we're going to use our length and our banding patterns to figure out what goes with what. We've got a color printout that's definitely much more modern, more representative of what you'll see in a lab today. Certainly you'll still see some black and white printouts, but you see we've got a male here because we've got an X and a Y. Notice how much shorter the Y is. That's again going to be really big later. And then just kind of a review of those terms. So each pair of homologous chromosomes includes one chromosome from each parent. Two sets of 23, one from mom, one from dad. We know all that kind of fun stuff. Diploid cell has two sets of chromosomes, so we should be good there. Haploid set, think half set. When you see haploid, it's going to be a single set of chromosomes. So that for humans, that's going to be 23. For corn, it's 10. For fruit flies, it's 4. Those are going to be the number of chromosomes found in the gametes or the sex cells. So gametes are haploid, so they get this N notation. Our haploid number is 23. Each set of 23 is 22 autosomes and a single sex chromosome. Kind of some more vocab there. In an unfertilized egg, the sex chromosome is X in the sperm. The sex chromosome may be either X or Y. That's why lots of people say the dad determines the gender. Polyploids are going to be entire sets of chromosomes. So we're not talking about a trisomy here. We're talking about a polyploid. So we've got an entire extra set. So instead of being diploid or haploid, now we're tetraploid or we're triploid. Really common in plants, but most of the time in animals, it's fatal. So here are some variations of the sexual life cycle. We see in animals, this is the one we're familiar with. Some plants and algae create these things called spores that may be around for thousands of years. They're essentially eggs that can last a really, really, really long time. And then some fungi and other protists have a slightly different version where this haploid unicellular or multicellular organism comes in to fertilize, gets some external fertilization. Plants and algae exhibit alternation of generations, so that's going to involve the spores and the sporophytes that I was talking about earlier. It's going to the life cycle as a whole is going to include both the diploid and haploid multicellular stage. So that's what we're seeing right here, and I'll provide some more information on that in a subsequent lecture. Okay, so in plants, each spore is going to grow by mitosis into a haploid organism called a gametophyte. The gametophyte is going to make gametes by mitosis, and then the fertilization of the gametes results in a diploid sporophyte. So that's essentially this alternation of generations that we're talking about. Like I said, I'll post some more information on that because I know I'm not doing a good job of explaining it. I'm not feeling well today, so I'm kind of stumbling through my words. But another variation, I talked through this one on the picture. That's that far right picture. The key is that there's no multicellular diploid stage. The zygote produces haploid cells through meiosis. During that, each haploid cell is going to grow by mitosis into a haploid multicellular organism. So there's no two sets. There's only one set of each chromosome. So differences of meiosis and mitosis. 2 cell divisions, not 1. We're going to produce 4 cells, not 2. The synapsis and chiasmata are going to be observed in meiosis. That's essentially crossing over, if you remember that term from Biology 1. Okay, 3 events are unique in meiosis, and they all are in meiosis 1. So synapsis and crossing over are going to be in prophase 1. That's where our legs are going to get stuck together, arms are going to get stuck together. Rather than untangling them, we're just going to cut them and glue them back together where they are. It's going to cause some variation. At the metaphase plate, again, remember in meiosis 1, there are paired homologous chromosomes, which we call tetrads, instead of individual replicated chromosomes. So we've got twice the DNA. And then at anaphase 1, the homologous chromosomes instead of the sister chromatids separate. So same thing, just more DNA. Continuing on our preview of differences, the first division separates pairs of chromosomes, not duplicate chromosomes. And then interkinesis is going to be present, so that's between meiosis 1 and meiosis 2. Meiosis is going to have 2 cell divisions, so it's going to be meiosis 1 and meiosis 2. All of our phase names will be anaphase 1, prophase 1, metaphase 1, and 2, all those types of things. I'm going to run through the steps really quick because they're essentially the same thing that happens during mitosis, but we've got twice as much DNA. So synapsis occurs as the chromosomes condense. Synapsis is where we're going to form our bivalents. We're going to form those tetrads. So our homologs are going to match up, basically. In prophase 1, again, we're going to see chiasmata, which is where we see crossing over. That's our longest phase of division, which should make sense because we've got the most DNA piled up in one place trying to sort it out. Metaphase 1, those bivalents and tetrads are going to align along the metaphase plate. Centromeres are going to point towards opposite poles. Anaphase, they're going to pull apart, but it's homologous pairs pulling apart, not individual chromosomes. Hopefully you remember this picture from Biology 1 that talks about independent assortment. If we get our 2 blue chromosomes over here on the left just by chance, we're going to end up with 2 sperm cells that look a lot like Dad, like Grandpa, and 2 that look a lot like Grandma. If the blue came from Dad's dad and Dad's mom. Over here in this possibility, we've got opposites. Just by chance, our blues are opposite one another. So this sperm cell is going to look a little bit like Grandpa, a little bit like Grandma. Same thing here. So we've got 4 different possibilities. They're all equally likely. In phase 1, the maternal and paternal chromosomes, now there's no difference between them. They just are chromosomes. Telophase, they're going to break up. It looks the same as it does during mitosis. So in interkinesis, no DNA synthesis occurs. This is between meiosis 1 and meiosis 2. May last for years, or the cell may immediately go into meiosis 2, depending on your gender. May appear similar to interphase of mitosis in terms of what's happening with the DNA and the chromatin and all those types of things. Meiosis 2 steps are the same: prophase, metaphase, anaphase, telophase. This looks a lot like the mitosis steps. However, the results are different. We have 4 cells produced that each have half the number of chromosomes. These cells are not exact copies of somatic cells, they're gametes. And then our genetic variation has increased drastically. That picture kind of summarizes all that. There's a table that summarizes all that. Comes straight out of your textbook, so I'm not spending a lot of time talking through those. 3 big sources of genetic variation in meiosis: independent assortment we talked through, random fertilization, and crossing over we'll talk through here in a few. So in humans, let's apply some of our math to the things to that picture I showed. We've got 23 pairs of chromosomes, so a chance to inherit 1 chromosome is 1 out of 2. You got a 50-50 shot. So think, I'm making the sperm cells for my children. My son has a 50-50 shot of getting my chromosome 1 from my dad, and a 50-50 shot of getting my chromosome 1 from my mom. That's what we're seeing here. Chromosome 1, chromosome 2. What independent assortment says is that these things are not linked. It's equally likely that my son gets my dad's chromosome 1 and my dad's chromosome 2. And it's equally likely that he gets my dad's 1 and my mom's 2. If we apply the math to that, there are this many possible combinations just from one person. So I can make that many different 8,388,608 different versions of sperm cells. My wife can make that many different versions of egg cells. Random fertilization, I really like this picture because it shows that fertilization is truly random. It's not a race. It's not the first sperm that reaches the egg. It's the first cell that's chemically compatible with the egg that gets in. Once that happens, all the doors lock, if things happen properly, and no other sperm cells get in. There are many stories of healthy couples that just can't have babies, and then they divorce or they break up or whatever, and they go their separate ways, and they each have children with other people that they are chemically compatible with. Tom Cruise and Nicole Kidman is an example of this. They both struggled for years to have kids. Then they split up, and it wasn't a very long time later before Tom had a baby and Nicole had kids. So, independent assortment, random fertilization. We'll pick up with crossing over in part 3. Hey guys, Mr. Hyatt here. This is part 3 of the meiosis lecture. So, we looked at a little bit of math in part 2. We said there are 8,388,608 different versions of sperm cells that I can create, and the same number of eggs that my wife can create. Put those together, that means that there are over 64 million combinations of my chromosomes and my wife's chromosomes. That kind of makes me feel, I don't know, a little bit special, like I'm one of 64 million possibilities that I could have been. It kind of makes you feel, I don't know, small when you see a number like that. So think about what that means in terms of your siblings. I mean, we look a little bit like each other, but we all look different, unless you're an identical twin. It's crazy that there are 64 million combinations that you could be, and that resets every time. So it's no wonder that we have some traits in common, but overall we look different. The thing that really throws a wrench into all of this math is crossing over, and makes this mathematically impossible to calculate. Those face combining apps and things like that you see sometimes on Facebook are just ridiculous, because number one, they can't account for the 64 million combinations that random fertilization ensures, and independent assortment ensures, but they definitely can't quantify crossing over. We'll do a lab that'll modify or model crossing over. That I think will help it make a lot of sense. But it's where we're going to have trade material. These chiasmata are points of contact. We see these chromosomes where they overlap. And I like to explain it like when you were a little kid and you fell asleep chewing gum. It's a lot easier for your mom to just cut the tangle out of your hair than it is to try to untangle hair by hair. And that's the same logic that our cells are going to use. So we see these chiasmata where we get some crossing over and we swap some DNA, so we end up with an all blue chromosome, a chromosome that's mostly blue with a little bit of red, a chromosome that's mostly red with a little bit of blue, and then an all red chromosome. So now we've got even more possibilities for chromosomes, especially combinations. It's going to break old linkage groups for sure. It's going to create new linkage groups that are going to increase variation. Really common during meiosis. Think about it though. It happens in prophase. Prophase 1 is when we've got essentially 4 copies of every chromosome. And they're all going to be piled up. They're going to stick to each other. They're going to be very sticky with one another. And so that makes sense that that's when it would happen. Notice that we can use the frequency to map the positions of genes on chromosomes. So think of it this way. When you were a little kid, you probably played Red Rover. If you are on the far left end and your best friend is on the far right end, you're probably going to get split up a bunch of times. That means you're not very close together because you've been split up so many times. If you and your best friend are side by side, you're not going to be split as many times. Every person that runs from the other side to your side isn't going to split you up. So you're going to be split up far less often, which implies that you're closer together. If you don't know your original position. I think that makes sense. So with crossing over, an offspring can never be 100% like a parent if we're using sexual reproduction. Because some of my chromosomes that I passed on to my son are partially from my dad and are partially, well, let me say that a different way. Some of them are even partially from my grandpa and partially from my grandma, because my dad's chromosomes crossed over in his prophase. So then I inherit a chromosome 1 that's partially my grandma, partially my grandpa, and then it crosses over with one that's from my mom's side, and now we've got all sorts of variation. Multiple crossovers are really common on large chromosomes. You see kind of what that would look like here. Smaller chromosomes, obviously we've got smaller surface area. genes near the centromere aren't going to cross over very often because they're tightly wound to the centromere. So just to summarize these three presentations, make sure you know how the chromosomes separate during meiosis. Make sure you have a mental image in your head of how those chromosomes move around. Know the terms, what we call them at each different phase. Are they a tetrad? Are they a homolog? It takes a little bit of practice with them, but you guys are all smart people or you wouldn't be here. Know the comparison between meiosis and mitosis. Be able to explain how each of those three things we talked about increases variation through sexual reproduction. That's meiosis.